Hello, everyone. I believe now we will start the neuroscience class. So just a quick overview of how it's going to happen. I'll be going through the slides, and at the end, I'm going to be giving some practice problems, and we'll have a Q&A session where you can ask me questions, and I'll ask you questions, and yeah. So yeah, without further ado, I think I'll get like a 1 or 2 minute break, or not 1 or 2 minute break, but like in case y'all want people need to like come back from the screens, and then we'll start soon. Yeah, just wait like a minute more. Okay, it's 7:01. I'll just start. Okay, share screen. Screen 2. Share. Okay, so this class, it's the first week for the neuroscience class, and we'll be discussing nervous tissue. So this is a brief overview. In this class for neuroscience, we're going to be going through a lot of things, but for this week, we will be discussing the main overall parts of the nervous system, as well as the tiny, intricate, and molecular details that people often overlook. For example, the cells of it and how these cells interact and communicate with each other so that the nervous system becomes its unique self. So this will be the first class of the nervous tissue. And let me go. Okay. So now let's go to the overall. Your nervous system is an extremely complicated system. It allows us to breathe, eat, talk, sing, feel, and pretty much everything our body needs to function. It works with other body systems and organs. For example, the heart to help it keep it functioning. Another example could be your muscles. For example, whenever you're playing soccer, so you can kick a ball. It does this by sending signals to these body parts, giving them instructions on what to do. So in the nervous system, there are two main subsections that people that study the nervous system or neurologists like to categorize. The first is the central nervous system. This is made up of the brain and a structure known as the spinal cord. As the name suggests, the spinal cord is a whole collection of nerves that run down your spine. The second main subsection is known as the peripheral nervous system. This is made up of all the nerves that go to your muscles and your different organs outside of the brain and spinal cord. For example, the nerves that go to your muscles, for example, to help you lift a rock, that would be the peripheral nervous system, or the nerves that could go to, for example, the different organs in your body, for example, like the stomach, and that would be part of that as well. So right here on the left, can I get a pointer? Yes. Right here on the left, we have the spinal cord. As you can see, this is your brain, and it goes down to the spinal cord, down the spine of your back. Right here, we have the brain, which I'm sure many of y'all are familiar with. And right here, we have the peripheral nerves. As you can see, the nerves extend from the spinal cord, and they go to the arms, legs, the arm, legs, and other places. So I'm going to give y'all a bit to, if you want to look at it, for example, to catch up. And then we'll move on to the next slide. Okay. Oh, oops. All right. So next we'll be going on. Going on to first is the neuron and its properties. The neuron is a cell that is unique to the nervous system. It is a microscopically small structure. However, it has an extremely important role in carrying all the electrical signals in your body from place to place. All neurons have three properties that allow them to communicate with each other. The first is that neurons can respond to changes in their environment. We call these changes in their environment as stimuli. and what it produces, or a stimulus. An example of this would be when you have your finger and you touch your arm. That would be a stimuli, and that would help, and the neurons would help respond to that by signaling that. Secondly, neurons can respond to changes by generating electrical currents that travel very quickly to cells far away. For example, if you want, let's do the soccer example again. If you want to play soccer, your brain would generate an electrical signal or a motor plan that would go down to your legs for you to kick the soccer ball. As soon as you think you want to kick the soccer ball, it's going to happen. It's really fascinating how the signals can travel from the top of your body to the very tippity tip bottom of your toes in such a really short amount of time. Then once the signal reaches the end of a neuron, the signal will be sent to another cell by a molecule known as a neurotransmitter. This will be discussed later. I'll give some time to go over this slide. Okay, we good? So here's how a neuron looks like, and we'll get into these structures later. Or actually, next, no, yeah. So next we'll be describing the different types of neurons. So there are three main types, or classes, as we say, of neurons in the nervous system. They are known as sensory neurons, interneurons, and motor neurons. Sensory neurons detect the environmental changes, and they help, or the stimuli, and they transmit this information to your brain. An example would be with the touching your arm or finger, for example. The sensory neurons, they pick up your finger touching this, and then they signal that on to the interneurons, which is the neurons that receive the electrical signals from the sensory neurons, and they send them to the motor neurons, described next. And the motor neurons are neurons that send the signals to your muscles, allowing you to move. We call them motor neurons because of the muscle function, but it also has functions in the different organs of your body. For example, the stomach or the kidney, or just being like any areas that these nervous systems areas is going to provide input to. Next, yeah, we can stay for that side a bit. Right here, you don't have to know all this stuff right now, but it's just saying that sensory neuron, when you get hit with a cactus, it traverses all the way up here, and then it goes to the interneurons and the motor neurons that allow you to remove your hand away. So, yep. Now we will be discussing the structure of a neuron. The neuron has many parts to it that can help transmit electrical signals throughout the nervous system. Three main structures are the cell body, the axon, and the dendrites. The cell body, also known as the soma, is this part of the neuron is the control center of it. Think of it as the brain, ironically, within the actual neuron. It contains a part of the cell known as the nucleus, which makes all the proteins that your cell needs. It also contains the DNA, or the inherited information about it. It also has the mitochondria, which is known as the powerhouse of the cell, generating the energy that the neuron needs to keep its functions going, because the neuron is always constantly active, or it's active for a majority part, and it's constantly firing like signals and electrical signals. So if you need to have a lot of energy, which is where the mitochondria comes into place. Next is the dendrites. The dendrites extend from the cell body. You receive the signals from the other neurons in the dendrites, and they look tangled and bushy, which is one way to help identify them. Now it's the axon. The axon extends from the cell body, but from the opposite side of the dendrites. They're usually cylindrical, and they conduct the information received from the dendrites. Neurons typically have one axon, while they can have many dendrites. And the axon, another important thing, that at the end of them, they branch into areas called axon terminals, which is where the signals are released from. See right here, cell body, the dendrite, the axon, this long cylindrical thing, and this is the axon terminals. So yeah, okay. If you want to look at this I'll give it there for a second. Okay. Let me. Oh, okay. All right. So now we'll be going on to another part of cells called the support or glial cells of the nervous system. So while neurons are extensively studied, there are other important cells in the nervous system. These other cells that aren't actually neurons are referred to as glial cells and support the neurons in numerous ways. So they don't actually send transmission and electrical impulses, but they rather help sustain the neuron and keep it functioning. The two main glial cells we will discuss are astrocytes and Schwann cells. Astrocytes, so they run a checkup on the brain, making sure there aren't any chemicals or any molecules that shouldn't be there. And then they remove those molecules if they are found. Next are Schwann cells. Schwann cells are pronounced, well, yeah, Schwann right there. And they produce myelin, which will be discussed later. The third type of cell in the nervous glial cell, and that is microglia, which I didn't really put here on the slides, but microglia are sort of like the immune cells, and they help prevent the nervous system from going under an attack from, let's say, viruses, bacteria, all of that. So I'll leave this here for a bit. Okay, let's go here. So yeah, here is a picture of an astrocyte. You can sort of see why it's called an astrocyte, because the word astro means star and cyte means cell. So it looks like these branches from the astrocyte right here look like multiple stars, really. So that's why it's called that. And this is the structure of a typical neuron, but you can find the Schwann cells right here, and they sort of envelope the axon, you could say. And yeah, right here along the axon's length. And you can see the myelin right here, the Schwann cell inside of it, that makes it. Okay. Now we'll go into the myelin sheath. So thing to know about myelin is that it surrounds the axon in some neurons, not all of them though, and it allows for the rapid transmission of these signals. Production of the myelin sheath is known as myelination, and the gaps between a myelin are known as nodes of Ranvier. The thing with myelin is the true function of myelin is really great and fascinating in how it can how can make neurons send these signals really quickly. Because without the myelin in neurons that we say are unmyelinated, the electrical signal, it can even if it's really good at the beginning, it slowly diminishes as time goes on without myelination. However, when there is myelin, the signal finds a way to regenerate itself and continues to go along the axon, which can allow for faster speeds and more efficient signaling. It can be really explained with an example of pain, how pain is transferred in your nervous system. So there's first type of pain, which we'll go over into this more later in another week. But this type is, let's say, when you hit your hand, you first feel an initial sharp pain. This is because the neurons that pick up the sharp pain have a lot of this myelin that allows for the nervous signals to reach your brain really quickly. However, there is also the slower pain that comes afterwards that can maybe last a few minutes or longer. This is because the nervous fibers or the neurons that perceive these signals don't have myelin, so it takes it slower for them to go up to our brain, which is why we perceive it later on. Okay. Now we'll be going on to the electrical properties of a neuron. So the neuron generates electrical signals and sends or propagates these signals throughout the nervous system. So one important electrical property we should be familiar with is current. Current is the flow of electrical charge from one point to another, from point A to point B. The currents and the charges of a neuron are determined by the amount of ions charged particles in and around the cell. The main ions that affect current are sodium ions, potassium ions, and chloride ions. What's really interesting about this is that all these things are here, sodium, potassium, chloride, or from chlorine, they all come from the periodic table, and they can all be used in our regular day-to-day things. For example, sodium and chloride together can make table salt. And the thing with this is it shows that all these different molecules, while they can have other functions, they can all be really important in the microscopic world that we never see and we never get to actually experience. And they're really important for that. In a neuron at rest, there are more positively charged particles, or as we call them, cations, on the outside of the cell. We call negatively charged particles anions as well. The differences in the two charges outside and inside the cell describes a property called potential. For example, if the, well, I'm going to be using a unit called volt, but that doesn't really matter. Let's say on the outside, it was 5, for example, the charge there, and on the inside, it was 2. So the potential across it would be 3. So that's what, because you had to subtract the 2 and find the difference between the two charges. And usually in the unit that they use is volts, because it's voltage. I'm sure you heard with the battery, because the battery might say it has a 5-volt battery, because that's describing its potential. So a lot of electrical properties do relate to the nervous system in this way. Next, we will discuss the generation of electrical signals. When you stimulate or disturb a neuron, there are disturbances in this potential described. This disturbance alters the balance of the amount of charged particles inside or outside the cell. For example, let's say I hit my arm, and then ions are moving everywhere because of the stimuli. What happens is, let's say on the outside of the cell, there are 5 cations, which is obviously a much lower number than it actually is. There are tons of molecules and ions that are involved in this process. So let's say there's 5 there, and on the inside, there are 2, or let's say it's 3 on the inside. After the disturbance, the movement of the ions gets transferred and it gets disturbed. So let's say after it's all said and done, on the inside of the neuron, there could also be 5 now. On the outside, there could be 3. So it could have just completely flipped in that sense. So that's how good a way to visualize how it occurs. Whenever more positive ions enter the cell, we say that we call that depolarized, what the cell state is in. Whenever more positive ions enter the cell. You don't really have to know the meaning behind it, but know that this is because, like, usually the cell, as we said, it's at negative, or it's at negative potential, right? So whenever more cations or positively charged ions enter it, then that means the amount of positively charged ions on the outside of the cell and on the inside of the cell are getting more similar. So this state allows for the electrical signaling to occur. Whenever the neuron is depolarized with more positive ions in, know that allows for more electrical signaling. So that's a good thing to draw there. If enough cations can enter the cell, an action potential will be generated. So an action potential is just a really strong electrical signal that can travel really long distances. And yes, they're traveled on axons as well. So now we'll be discussing the spreading or the conduction of these signals. So actually, I'll leave this up here for a bit in case anyone wants to take notes. Okay, that should be good. So spreading and conduction of the signals. So most potentials spread down the axon, and electrical signals start in the beginning of the axon around right here. We call this area that it starts in the axon hillock, and the electrical signal first starts in an area called the initial segment in that area. So just prefer to say it's the initial segment. So the action potentials, they regenerate themselves along the axon so they can traverse the entire length. This is because, for example, this right here is a neuron at rest. So you can see there's more positive outside it and negative inside. So what happens is, with the action potential comes, the positive ions come inside the cell, and this leads to the positive ions right here to go inside, which then leads to the positive ions here to go inside, here to go inside. So then it's just a constantly going down signal. So yeah. And then the neurons travel faster with myelin because of the special properties of myelin, which mean that the myelin allows for more of these signals to regenerate in that case. So yeah, with myelin, it allows for more regeneration. So that's like the main reason to why the myelin actually allows for faster signaling, is because they allow for better regeneration of the signal along the axon. Okay. Now, next. So now we'll be discussing synapses. The place where neurons interact with each other is known as a synapse, and the space between them, whenever they interact, is known as the synaptic cleft. The neuron that is sending the signals to the next neuron is called the presynaptic neuron. And no, that's called presynaptic because pre means before, then synapse means synapse. So it's before the synapse. So think of it right here, and the synapse is right here. And then the neuron that's receiving the signal from the presynaptic neuron, or the sending neuron, is called the postsynaptic neuron, or the receiving neuron. The presynaptic cell releases chemicals known as neurotransmitters to signal to the postsynaptic cells. So keep in mind with the so these three terms are really crucial, as presynaptic, postsynaptic, and neurotransmitters are pretty prevalent terms in neuroscience in general. So knowing this a lot, and we'll get into neurotransmitters in a bit later, and like different examples of them and how they work. Okay. Let's give it a second right here if anyone wants to take notes, maybe. Okay. Now let's go into the sending neuron. Or no, yeah, so sending neuron right here. And you see, you will learn a couple more of these other terms later, but you see the synapse and you see the neurotransmitter, sending neuron, and that's how it mainly looks. We'll look back at this diagram in a bit after this slide. The neurotransmitter slide. So there are more than 100 different neurotransmitters that have been discovered. And so yeah, most of these are small molecules that are stored in vesicles, known as vesicles in the presynaptic cell. Think of these vesicles as small bubbles, as you can see right here, that hold the neurotransmitter and release them. And they're released by a process known as exocytosis. I won't get into the specific details of exocytosis, but suffice it to say right now, that's just how the vesicles release a neurotransmitter. These neurotransmitters will bind to receptors, which just bind to the neurotransmitter and elicit a response. You could see the receptors right here in the neurotransmitter. So here's a really key thing about this. Even though a neurotransmitter, for example, typically does an action, for example, neurotransmitter A in most neurons will cause B, or an effect of B. It is the action of the receptor to which the postsynaptic cell is affected. So if the receptor wasn't working, even if the neurotransmitter is released, no effect will occur, because the receptor is sort of like the relay that carries this information and causes an effect to happen in the next cell. So the receptors are really key to determining the function of it. Let's say that the receptor as well could have been, let's say the receptor as well couldn't have been functioning, and causes a different response that shouldn't be happening, then it would be causing something to like, yeah, not happening. So someone asked, I'll just answer it. Are neurotransmitters proteins? So I'll press answer live. So some neurotransmitters are proteins and some aren't proteins as well. For example, there are different classes of neurotransmitters. One could be protein, for example, whereas other neurotransmitters could be small molecules, such as acetylcholine, epinephrine, serotonin, and glutamate. Acetylcholine is a neurotransmitter that goes to a neurotransmitter that causes, let's say, your muscles to move. And epinephrine or adrenaline, as you probably would know, causes the flight or fight response. So, but another thing is proteins that are active neurotransmitters act as a different subset of neurotransmitters called neuromodulators, which we will get into in the next slide. Okay, someone asked, what are proteins? So proteins, think of it as large molecules that large molecules that are important to the cell. For example, in your cell, they could build up in your cell, right? Like they could cause different actions in your cell, and they're really big, or like they're bigger molecules. For example, some proteins can make chemical reactions go faster in these areas. Oh, what is a cell? Okay, so that's fine. So a cell is basically the basic unit of life. So let's say on a human, right, that we have skin cells. So these skin cells carry out the functions of what we need to do to make up our skin. So say that a cell has a nucleus, like we said, and this nucleus helps carry out the information and the different functions of that cell. So yeah, I'll press and yeah. Okay. So next is the end of the signal. So after the chemical signal gets sent to the next neuron, the signal needs to end, obviously, because if the signal didn't end, then it would just keep going on, which shouldn't be happening. But how does this actually occur? One way is through proteins known as enzymes. So if you don't know what that is, a protein is just a large molecule, think of it like that. And the enzyme is a type of protein that makes certain chemical reactions go faster. For example, let's say if A to B, we're trying to get molecule A to go into substance B. In a normal environment, this would take a really long time. However, with enzymes, they take a really short amount of time. So, and that's how it breaks down the neurotransmitter. Once it is broken down, it will no longer have an effect on the receiving cell because it can't go to the receptors. The second way is by sending the cell back to the neurotransmitter, which is like the neurotransmitter gets reuptaken by the actual cell. This is accomplished by special transport molecules that allow for the neurotransmitter to go back into the cell. And so you don't have to know everything right here, but right here you see the enzymes, they inactivate the neurotransmitter. So you have an active neurotransmitter right here. After it goes to an enzyme, it becomes a broken down version of itself, or could it just go back in? We say another way that we didn't really talk about, which is that it just leaves the entire synapse area, which is one mechanism that works. And then finally, we'll discuss neuromodulators. So these are included under the umbrella term neurotransmitter, but they have somewhat a different function. If you were to think of neurotransmitters as the cells that cause signals to happen, think of neuromodulators as the different molecules that sort of tweak the response of the neurotransmitter. For example, they could prevent the neurotransmitter from ever being removed, thus making the signal last longer. On the other hand, it could also decrease the amount of neurotransmitter even released, thus blocking the entire signal. And it can also make the receiving cell more sensitive to certain signals. So yeah, which causes a bigger response if it ever does bind or go to the receptor. A notable example of a neuromodulator is endorphin. endorphin is really important. It blocks the sending or the transmission of pain signals to the brain and spinal cord. We'll go over this in a later week, but right now think that, let's say you really hurt your hand. After a while, the pain starts to numb away, and that's mainly due to the action of endorphin, which this molecule, which blocks it, which blocks the pain signals. And that's how it works as a neuromodulator. Okay. So yes. All right, so we'll start the other stuff now. But so thank you all for listening to this long lecture. And I hope it wasn't boring and it inspires you all to learn more about the fascinating properties of neurons. If you have any questions or you critiques about it, please tell me. And yeah, so we'll have a Q&A session. And then after that, if we have time, I'll ask some questions about what we learned. So yeah, go ask ahead. Any way to obtain intuition for this? I don't know what you mean by that. Like, what? Could you please elaborate what you mean by that? Any way to obtain information? Yeah, actually, yeah. Could you just elaborate that? I don't really understand your question. Because, I mean, intuition is just like you being able to solve a problem you don't know based on the previous information you had. So, I mean, if you get previous information, then you get intuition. I don't really understand that question. But yeah, continue. Continue to ask as much questions as you want. Also, if you want to give a critique, like maybe if I went too fast, too slow, or if the stuff was maybe too hard or too easy, yeah, please go ahead and give that. I would definitely appreciate that as well as questions about certain parts. How do neurons communicate with each other? Okay, so let me scroll up right here. The neurotransmitters, we said, actually, I don't really need to. So yeah, so these neurotransmitters that are released, they go bind to the actual receptors. And when they bind to the receptors cause a different effect in the next neuron or the postsynaptic neuron. And that neuron effect, basically, that's how the neuron signals are communicated, because it sends the signal in the form of a neurotransmitter to another cell or a neuron, and then that carries the signal now. So that's how they communicate. It's sort of, think of it as the electrical signal just jumped from one neuron to the next neuron, and that goes down. That's a great question. So yeah, continue asking questions. Where are neurotransmitters stored before they are released? So as you can see right here in this picture, they're stored in these vesicles in the presynaptic or the sending neuron. So yeah, that's where they're stored. How do neurotransmitters travel? So these neurotransmitters, once they're in these vesicles, as you can see right here, they're released into this fluid that's outside the cell. So you might think, okay, well then how does it bind to the receptor maybe? The thing is, the next cell is usually so close, like with microscopically close distances to the sending neuron and the receiving neuron, that the neurotransmitter can the neurotransmitter, right? It has a high chance of binding to receptor almost immediately in that sense. So they travel from this inside of the cell and they go to this area in the synapse in between the two neurons. Then by then they go to the receptor. And this can happen mainly by a process known as diffusion, where the molecules spread throughout a cell, or not a cell, but spread throughout like a liquid, for example. And so yeah, that's how they travel. Good question. Yeah. How do the electrical signals travel through neurons so fast? Well, the answer with that lies in the myelin that we went about. Let me. So the myelin sheath. And right here, the myelin, what it does is it allows for the signals to regenerate along the axon. as we said. And so this, what it does is it, myelin axons to go along, and when it regenerates, it can thus go down really fast and much more faster distances because it allows it to conduct faster. So good question. What is the half-life of TNF transforming a tumor necrosis factor alpha in vitro? That's not anything pertaining to what we are doing. And that seems like a pretty random question to ask. So I wouldn't know that. You can probably look it up. And if you're knowing stuff like that, you probably already know stuff. So yeah. I don't know, but so yeah, does the myelin production decrease throughout age? Yes, it actually does decrease throughout age, and we'll be going into some certain diseases in a later week about what happens. And so that's why the nervous system function actually gets lower with age. What is the function of a dendrite? So a dendrite, what it does is it picks up the signals from another neuron. For example, right here, this right here in the receiving neuron is most likely going to be the dendrite. And when the dendrite happens is it receives the neurotransmitters and then it sends the signal to the axon and the cell body. Good idea. How many neurons are in the brain? I'm pretty sure there are 100 billion. So yeah, 100 billion. Did I say disease? Yes, we will be going over certain diseases about the nervous system because these we'll be learning about them. And we'll mainly be learning about their specific neural mechanisms because they are pretty important. Because when you have when you learn about the mechanisms in part of the disease, then you can allow different functions. For example, a lot of times different scientists, when they had a disease, they knew there was a malfunction in a certain structure. And because there's a malfunction in that certain structure, they could figure out what that structure normally does in a normal person state. That's a good thing. How many other types of tissues work like nervous tissues or like work with nervous tissues? So almost all tissues actually receive the nervous input. In a tissue, if you don't know, it's just like a bunch of collections of cells in your body. For example, your heart tissue receives input from your nervous system, your muscle tissue, your stomach, and different all throughout your entire body, your skin. Is the cause of Alzheimer's disease from neurons malfunctioning? Yes, it is the cause of neurons malfunctioning in that sense, which can cause, we see like the memories part with it. But there are other more intricate parts of the Alzheimer's disease which cause the neurons to malfunction, such as the buildup of toxic proteins. How to differentiate between dendrite and axon? So if you go right here, you can see the dendrites are right here and the axon is right here. So the axon, what it does is, I think of the axon as one long cylinder. In that sense, that carries it down. And the dendrites receive the signals from other neurons. If you're talking about that sense, so dendrites receive signals from other neurons, while the axon receives the signal from the dendrite of that same cell. And the dendrites look a lot more bushier, and the axon is like one single thing going down. Generally, what do prions do to your brain? Okay, that is not what we are covering in this class. So it would be better like if you ask a question, maybe that could like attend to it. But yeah, because the prion, that's pretty a bit too advanced for this. So I won't really get into that. But yeah, you can look it up, I guess. It's a bit too advanced for this. I don't want to confuse people. Do the axons include the Schwann cells and the myelin sheath? Yes, that's what happens. So the Schwann cells actually make the myelin sheath. And you can see right here, they sort of like wrap around the axon. So that's why you see the Schwann cells and the myelin sheath are pointing to the same thing. Because like the Schwann cell, it goes to attaches to an axon, and then it wraps around it a bunch of times, and then it makes the myelin, which then allows for the cell to go faster. What do Schwann cells do? So Schwann cells, as we said, I just said, but they make the myelin, and this myelin can later on help the nervous system get faster. So yeah, great questions. How are you? So, okay. Okay. If there are any more problems people have or questions, any critique as well would be valued about it. Like what? So yeah, what did y'all think as well? Oh yeah, what does specifically do the nodes of Ranvier do? Okay, so these things I didn't really get into it. I didn't want to go too much about it. But the nodes of Ranvier, they are important in the regeneration of it. The nodes of Ranvier and the myelin work together to make it faster, is what it does. Yeah, it just works together with it. Yep. What happens if astrocyte is malfunctional? So if the astrocyte is malfunctional, this allows for certain toxic substances to get into the brain. Because recall right here, whenever we talked about astrocytes, yes, what they do is they make sure the molecules that should be there are there. So for example, if your astrocytes aren't working, then certain molecules, right, that are toxic could get into your brain. And if that's there, then it could cause a bunch of nervous system malfunction where neurons could just die in that things. What do cations and anions have to do with neurons? Well, it's fine if you forget. It was a lot of it packed into a 30-minute thing, so don't worry about it. So basically, the movement of, I'm trying to find it, but so the movement of the cations and the anions in your cells basically cause a current in your neurons. And this current, because they're charged, and each cation carries a charge, an anion. And when they carry this charge and the different movements happen, it carries a signal down your neurons based on their movements of them. Because what it does is it changes the potential, as we said, or the differences between the outside or the inside of the cell in terms of the charge, in terms of the charge. Good question as well. So, yeah. I don't think we'll have time to get into practice problems, so I think we'll just answer more questions. Probably be better. Yeah, I know. It's fine if you forget stuff, because I know y'all probably had a pretty long day with a bunch of different classes, and this is the last one. So is dementia the same as Alzheimer's? So dementia, no, they're not the same thing. We'll get into that more in the last week, but they're not the same they're not the same disease, but dementia is just like sort of like symptoms, I guess you could say, or something that, or like a disorder. But there are like two different things. And like dementia is sort of like a set of symptoms that you would get. Alzheimer's is like a set disease. Could you show me the first slide? Okay. Dementia does affect your memories. Yeah, it does. But you can have dementia, I'm pretty sure, without having Alzheimer's. The first slide. Wait, are you talking about the first slide of the overall information? Or, yeah. That's a fishbone diagram. No, this is your spinal cord. It does look like a fishbone, I will say that, but this is your spine, or your spine bones, and the nerves, they go along that. So yeah, on the overall info. Okay, yeah. Well, class is going to be ending soon, but yeah, just as you take that. Yeah. Any other last questions? Why does the head look so weird? Oh, does a neuron also exist in the spinal cord? Yes, neurons do exist in the spinal cord. So yeah, neurons do exist. Why does the head look so weird? This is just a different version. Like this is just like a different view of how it looks. So it's going to look a bit different. a lot of different diagrams look pretty weird, especially in anatomy when you go there. So yeah. Also, I think that would be the end of the class since it's 7:45. So what? Okay. Yeah, I think this is probably a good place to end it here. And the next week, we're going to be going into the brain and the spinal cord, I believe. It should say on the website in case I'm wrong. And yeah, I hope y'all had a, oh, show me the next slide. Okay, sorry. Yeah, I'm going to be posting all these slides in a Google Drive for everyone. So yeah. We could stay here for a little bit longer, but other than that, class is pretty much over. So yeah. Oh yeah, no problem. I hope this allowed, or hope this inspired all of y'all to maybe consider learning more about the neuroscience in addition to what's in this class. And so yeah. Yep, no problem. And I think that will be it. So do I have permission to end the meeting right now? Or so yeah. I'm pretty sure right now you have to end the meeting. So I'm gonna stop the screen share. So yeah, unless there's any other questions people have.